one of your two favorite topics in biology, right? And didn't you guys love metabolism in 150? No? You know, well, we are going to review a lot of the basics, but we're not going to go into the same detail that you did in 150. And we're also going to focus more on the prokaryotes next week when we finish this lecture. But to get started, metabolism is the sum total of all of the chemical reactions in an organism. And you can split those reactions into two major groups, catabolism and anabolism. And the catabolic reactions are those that break down food molecules and produce energy. And anabolism uses that energy for biosynthesis to build larger molecules and to do work. And what's the kind of energy we're talking about?
Is it ATP?
Yep, ATP. So once again, organisms consume energy in the form of food molecules, but they must convert that chemical potential energy in the food into ATP. So the catabolic reactions break down those food molecules and produce ATP. The anabolic reactions use that ATP for biosynthesis to build larger molecules and to do work. In the process, heat does get lost. In fact, the majority of that chemical potential energy in a food molecule is actually lost as heat. So cells perform metabolic pathways, so a series of enzymes which ultimately produce an end product. And so we are going to review enzymes and the factors that affect them. Although you should already be familiar with those, we are going to review it. But enzymes allow for cells to make things that they need, and they make reactions happen faster. And of course, enzymes are made of protein, so they are encoded by the genes. Now, as I'm sure you remember, all molecules are in motion. Even the molecules in the tabletop where you're sitting right now are in motion, just very, very slowly. But we are talking about biological reactions, so those are occurring in an aqueous environment, in a watery environment inside the cell. So you have a lot of molecules just randomly moving around, and occasionally they collide into each other. If they collide with sufficient energy, they will react and form a product. But most of the time, they just hit each other, bounce off, and go their separate ways. The amount of energy needed for those molecules to react is called the energy of activation, or the activation energy. Now, there is a way to increase the likelihood that those molecules will react to each other when they collide. And so, if you're working in a lab, what's sort of a simple way to make reactions happen faster? So what's a way to speed up reactions without using enzymes? Make those molecules move faster. How would you do that? Right, heat it up, right? Increase the temperature, and that gives those molecules more energy. The problem with that and life, especially complex multicellular organisms, is that the amount of heat needed in order to make these reactions happen fast enough for life to exist would basically be too hot for life to exist. So you can increase the rate of reactions by increasing the temperature of the environment, but that doesn't really work for living organisms. So they use enzymes. So what we see here is the normal amount of energy needed for reactants, right, the two molecules that are going to react together to form products. So the amount of energy needed for these reactants to form products. What enzymes do is they lower the energy of activation. And that allows the reactants to form products much faster. And that's fast enough for life to exist. So as these molecules are randomly colliding, they would need this amount of energy in order to react and form products. That's normally not available, at least not very often, in the environment. So enzymes lower the amount of energy needed for the reactants to form products. And so reactions happen fast, and that allows life to exist. So enzymes are biological catalysts. So a catalyst is something that makes something else happen. And the enzymes are substrate specific. So that means that only one particular substrate fits into the active site of that enzyme. And as hopefully you recall from Biology 150, that all has to do with all the chemical bonding and interactions that occur, right, between molecules. Hydrogen bonds, van der Waals forces, ionic bonds, all those kinds of things are what make the active site of an enzyme specific for only one kind of substrate, because that substrate will fit to the active site and also be able to make all the necessary chemical interactions. The enzymes are not permanently altered during a reaction. So by the time the reaction is over, the enzyme is exactly the way it was before the reaction began. So that way the enzyme can catalyze another reaction immediately after the first one, and over and over and over again. Enzymes are made of protein. Many enzymes require cofactors and coenzymes. So when you talk just about the protein portion, we call that the apoenzyme. And the prefix apo means it's missing something. And what it's missing is the cofactor or coenzyme needed to make it whole or complete, the holoenzyme. Cofactors are typically derived from inorganic minerals like iron, and manganese, and zinc. Coenzymes are typically derived from vitamins, especially the B vitamins. You may have heard that if you're feeling fatigued and tired all the time, that maybe you need some more B vitamins because they give you energy. Well, the B vitamins don't give you energy, but maybe all the enzymes needed to make ATP don't have enough of their coenzyme. And so by taking the B vitamins, you supply those enzymes with the coenzyme they need to be functional so you can make a lot more ATP. So here we see the apoenzyme, the protein portion. When it gets its coenzyme or cofactor, it becomes complete, it becomes whole, right, the holoenzyme. Now the substrate can bind to the active site. And the old example that was always used was the lock and key example, where only one key fits into a particular lock. And if you knew anything about locks, you knew that wasn't entirely accurate, but the whole point is that the active site, only one type of substrate can fit into that active site. Now you also notice there's an alternate binding site here. We're going to talk about that later, but that's not all enzymes have it, but some do. And that's used for regulating them, as we will see. Now, I'm sure you've all seen these letters before, and they probably scare you or make you feel kind of nauseous, because you are going to see some of these again. But things like NAD+, NADP+, FAD, coenzyme A, they do have many important roles inside the cell. And one of the most important roles is as an electron carrier. So all these coenzymes are very important electron carriers, because as we're going to see, it's all about the electrons. So enzymes make reactions happen fast, maybe as many as 10,000 molecules per second. An enzyme like DNA polymerase, which makes DNA, is able to add those nucleotides. Remember the A, T, Gs, and Cs? The DNA polymerase can add all those nucleotides at a rate of approximately 1,000 nucleotides per second. So that means every second, 1,000 ATGCs are added. So that's incredibly fast. But when you think of eukaryotes and those very long linear chromosomes they have, you have to replicate quickly. in order for that cell to divide. So you need an enzyme to speed that process up. Now, most enzymes end with ASE. So that's the standard naming convention, is using ASE. And the name usually indicates the function. So for example, a phosphatase removes phosphates from a molecule. A polymerase, like DNA polymerase, adds nucleotides to make polymers. And polymers are long chains of subunits. Amylase, you would need to know what amyl refers to, and it refers to sugars or carbohydrates. So amylase breaks down starch. Protease, that one's pretty easy, right? Enzymes that break down protein. Now, ligase is hard because you would need to know what the word ligate means, and most people have never heard of ligate, but ligate is to join together. So a ligase joins molecules together. And many of these enzymes do require ATP in order to function. Now, there were certain enzymes that were already known, they were already experimenting with them before they started using ASE. And does anybody know any examples of enzymes that do not start with ASE, or end with ASE rather? Most of these are proteases, but an enzyme that does not end with ASE. Nobody knows? Does anyone like to eat pineapple, fresh pineapple? Nobody likes pineapple?
I like pineapple.
If you were to eat a lot of it, what you would notice is that your gums start to get very tender. And the reason why is there is a protease in the pineapple. And when you eat a lot of it, it starts to attack your gums. And so they feel kind of raw and tender. And that enzyme is bromelain. So bromelain is an enzyme in pineapple. And a kiwi. I don't know that kiwi. I don't know if kiwis have anything in them or if they're just acidic. You have a question, Christiana?
Yeah, please, what's the correct spelling of gomelin?
Bromelin.
BROM.
ELIN.
E.
Right, so bromelain. There's another one. There's trypsin, chymotrypsin, pepsin. So those are enzymes that were already known, before. And so those names still stay the same. But new enzymes all end with ASE. Now, since enzymes are made of protein, there are 4 levels of protein structure. What are those 4 levels of protein structure?
Primary, secondary, tertiary, and quaternary.
Yep. And what is the primary level of protein structure? What is that?
Is it just the line of amino acids?
Right. It's just a sequence of amino acids. There are 2 secondary structures. What are those? 2 secondary structures.
Is it the alpha and the beta?
Well, the alpha what and the beta what?
The alpha amylase?
No.
No.
A green? No. We see it right here. This is the alpha what?
Oh, the helix. Alpha
Helix.
Alpha helix and the beta what?
Sheet.
Sheets,
Pleated
right?
sheet. Yeah.
Beta sheet, yep. And the tertiary structure is when everything folds on itself to form that 3-dimensional globular structure. And for many proteins, that's it. They're done, right? They're functional. But some other proteins require multiple subunits to come together, and that's the quaternary level of protein structure. Now, if you denature a protein, for most proteins you have destroyed them, right? They cannot renature, for most proteins. Some can, but most, once you denature them, they're destroyed. And the two ways to denature proteins are temperature and pH. And for temperature, I always use the example, once you fry an egg, you cannot put it back in the shell, right? Because you've denatured it, you've destroyed it, you changed those proteins. So here we have a temperature curve, and it's showing the enzyme activity. It's basically like a bell-shaped curve. And this point here represents the optimum temperature. So the temperature at which the enzyme works the best and the rate is the fastest. Now, if you heat the enzyme above the optimum temperature, the rate goes down, the enzyme becomes denatured. What if you take that denatured enzyme and you cool it back down to the optimum? Will it function?
Oh,
No, right? You denatured it, you destroyed it, so it's not going to function. But what if you take your enzyme, right, that's working, and then you just cool it, you chill it? What happens to the enzyme?
does it coagulate?
Nope. What happens to the enzyme, right, the activity?
It weighs more.
Well, the rate.
Oh, the
How
rate.
fast it works. Yeah, how fast it works. When you cool it, what happens to the rate? Right, the rate goes down, right? It slows down. And so what if you take your chilled enzyme and you warm it back up? Will it work? Right, so you take your cold enzyme and warm it back up. Does it work? Yes, it sure does, right? So chilling an enzyme only slows it down. So what that means for things like bacteria, refrigeration and freezing does not kill most bacteria. So when you refrigerate or freeze bacteria, it just slows them down. When you warm them back up, they work just fine, right? They start to multiply. Now the pH curve is another bell curve. Every enzyme has its own optimum pH. Now for most enzymes and most organisms, the optimum temperature is a moderate temperature, not the extremes. And the optimum pH also tends to be a moderate pH, usually around pH 7. But this does depend on the organism and the specific enzyme. Some enzymes work best in a more acidic pH, some in a more alkaline pH, but most tend to work best right around 7, but it depends on the enzyme. And in this case, if you decrease the pH, which increases the acidity, or you increase the pH, which increases the alkalinity, you will denature the enzyme and destroy it. So either too low a pH or too high a pH will denature the enzyme and destroy it. So enzymes all function within a narrow pH range as well as a narrow temperature range. Now, substrate concentration is a little bit more complicated. The idea here is that the amount of enzyme is fixed, right? So we have a fixed amount of enzyme. You start adding substrate and the rate of reaction increases. And as you add more and more substrate, the rate increases and increases and increases. You keep adding substrate, the rate increases up to a certain point, and then adding more substrate will not increase the rate of reaction. Why not? When you add substrate, the rate goes up initially and goes up and up and up. You keep adding substrate, but eventually the rate levels off. Adding more substrate will not increase that enzyme activity rate. Why not? What does that mean to reach an equilibrium in this case?
Is it that the reaction is happening just as fast as the things are being broken down? I don't know.
Well, you're halfway right. So the substrates bind to the active sites of the enzyme. Since the amount of enzyme is finite, is limited, so is the number of active sites. So as you increase the substrate, eventually all the active sites are full. The enzymes are working as fast as they can. Adding more substrate will not make the enzymes work faster, right? Because they're full, right? They're saturated. So does that make sense? Is it kind of like when your professor has more and more homework to do and you're already saturated and can't do any more? Right? Giving you more homework is not going to make you work faster, right?
Right.
Lucky for you, I don't do much homework. So now we can inhibit enzyme activity. And we're going to talk about the two types of inhibition, competitive and non-competitive inhibition. But first is competitive. So in this case, there is a molecule that resembles the substrate. And so the competitive inhibitor can bind to the active site of the enzyme. If the inhibitor is in the active site, the actual substrate cannot bind. So this enzyme has been blocked. So you can use a substance that resembles the actual substrate, and that substance will bind to the active site and block the actual substrate. How can we use this concept to kill bacteria? Right, so you want to make a lot of money, so use the concept of competitive inhibition. How can you kill bacteria that way? So who wants to get rich? Come up with an idea. You have an idea, Christiana? Roxy? Craig? Bryant?
Hello?
Gary? Yeah, Christiana?
Yeah, I said in the competitive inhibitor or something, people will have to buy more bacteria to get rid of, more antibiotics to get rid of the bacteria.
Right,
In
so
that case, we are selling more to make money.
right. Yeah, so the idea here is you need to find a critical metabolic pathway. So maybe one where the bacteria are making a product that they must have to survive, and they cannot absorb that product from the environment, they have to make it. And so you find that pathway, and then you find a chemical that resembles the substrate that you can use to block the active site and keep the substrate from binding. You just have to use enough of that chemical for a long enough period of time that it will block the active site and keep those bacteria from making that critical product, and then they die. And so that's how certain antibiotics work. So an example of that is the sulfa drug, sulfanilamide. So sulfanilamide is one of our sulfa antibiotics. It resembles PABA. So this is the substrate that many bacteria use to create folic acid or folate, an essential vitamin. Human beings do not synthesize folate. We get it from the foods we eat. But many bacteria, and especially certain pathogens, they must synthesize their folate. So you can use sulfa drugs to block the enzyme and prevent PABA from being converted into folate or folic acid. And so this is an example where sulfanilamide is a competitive inhibitor of PABA. Now, has anyone taken or are you taking organic chemistry right now? Nobody's done orgo? In the past. What about you, Roxy? Weren't you taking some chemistry? Okay, Taylor's taking it. So, does anyone know what PABA stands for? I mean, can you name this molecule? Oh, next semester. So, let's have a little organic chemistry lesson. So, this ring structure here is a benzene ring. This is a carboxyl group, which makes it an acid. So this part here is benzoic acid, and NH2 is an amino group. Now, when it comes to side groups like this, there is a way to name their orientation on the ring. And so they use the words ortho, meta, and para, as an ortho, meta, and para at this position. So PABA stands for para-amino benzoic acid. So what you guys have to look forward to if you're going to take organic chemistry. So PABA is the substrate bacteria use to make folate. Sulfanilamide blocks the active site of the enzyme and prevents those bacteria from making folate. And so eventually they die. So that's an example of competitive inhibition. Now, non-competitive inhibition involves enzymes that have this alternate binding site. We call this site the allosteric site. Not all enzymes have an allosteric site, but very often enzymes in metabolic pathways that produce a product do have an allosteric site. And so here is an example of where organisms can regulate their own metabolic pathways. So let's say, for example, the cell or the organism has made enough of a particular product. It does not need to make any more product. So if the cell or the organism continued to make product that it did not need, what would that waste? Energy, and in particular, microorganisms cannot afford to be wasteful. So once the cell has made enough of a particular product, that product can actually feed back to an enzyme early in the pathway, bind to the allosteric site. Right, so the non-competitive inhibitor is actually product, which binds to the allosteric site. That changes the shape of the enzyme, which changes the shape of the active site. So the substrate can no longer bind. So this enzyme has been turned off. Now the binding of the end product to the allosteric site is reversible, and it's concentration dependent. So the more of the product you have, the more likely it will bind to the allosteric site. As the cell consumes the product, as it uses up the product, it comes off the allosteric site, the enzyme goes back to its normal shape and starts to function again. Right? So the cell can make more product. So cells actually do regulate their own product formation. And that process is called feedback inhibition. So the end product, also called an effector molecule, when the cell's made more than enough of it, it feeds back to an enzyme, usually early in the pathway, because the earlier you turn off that enzyme, the more energy that can be saved. And so it binds to the allosteric site, changes the shape of the enzyme, which changes the shape of the active site. Now the substrate cannot bind. So the pathway has been shut down. Once the cell consumes the product, uses it up, then it comes off the allosteric site. The enzyme goes back to its normal shape and starts to function so the cell can make more product. And we already said, why does the cell want to do this?
To save energy.
Save energy. So here we see an example of a metabolic pathway which has more than one enzyme. So the end product, when it is in excess, can feed back to an enzyme, usually early in the pathway, right? And it binds to the allosteric site and changes the shape. So now the enzyme can no longer bind to its substrate. Normally this enzyme would bind substrate, convert that into a product, which then becomes the substrate for the next enzyme. And that enzyme converts that substrate into a product that becomes the substrate for the next enzyme. And so ultimately the last enzyme produces the final end product. But if you block an enzyme early in the pathway, there's nothing for these other enzymes to do. And almost all of these biosynthetic reactions require ATP. And so a lot of ATP gets saved. As the cell uses up the end product, it comes off the enzyme because the binding of the non-competitive inhibitor of the end product to the allosteric site is reversible. It comes on, it comes off, come on, come off. And the higher the concentration, the more likely the end product will be bound to the allosteric site, right? Because the binding is concentration dependent. So as the cell uses up the end product, it's no longer available to bind to the allosteric site. And so the enzyme goes back to its normal shape. And cells do this all the time with all different kinds of pathways, including the pathway that makes ATP. So ATP can also be an allosteric inhibitor and slow down the production of ATP inside a cell. Although most of the time organisms do not make excess ATP, but when they do, that organism can slow down the production of ATP. So all kinds of molecules can serve as effectors or as non-competitive inhibitors. So does that make sense?
Non-competitive inhibitors can be a good thing. They're not necessarily
Oh, yes,
trying
yes.
to.
Yep, cells do this on purpose.
Okay.
So now that you understand that concept, can you think of a way to use a non-competitive inhibitor to kill microorganisms? Any ideas?
Can it be chemotherapy?
What was that?
Is it chemotherapy for killing cancer cells?
Oh, chemotherapy. Oh, well, potentially. But can you give me some details as to how your therapy would work?
Shuts the pathway for the cells to stop growing.
And so maybe you find, right, a metabolic pathway needed for growth and reproduction, and you find a chemical that binds to the allosteric site, right, to stop that. Right, so there are different things that can be done. The idea is find a critical pathway, find the enzyme early in that pathway. If it's an allosteric enzyme, see if you can find a drug, a chemical that will bind to the allosteric site and change the shape of that enzyme. And then you can either kill microorganisms, or in the case of viruses, you don't kill them, but you might be able to stop them from replicating. An example, HIV uses the enzyme reverse transcriptase, which is an allosteric enzyme. So we have created drugs which bind to the allosteric site and change the shape of reverse transcriptase, thereby turning it off. And that prevents the viruses from replicating. But you do have to keep taking that drug every day for the rest of your life to keep the reverse transcriptase inactive. So there are ways that we can exploit this natural process that organisms use to regulate a metabolic pathway. And potentially you can make a lot of money. So now back to some basic chemistry, oxidation and reduction. As you should recall, oxidation is the loss of electrons from an atom or molecule, and reduction is the gain of electrons, right, by an atom or a molecule. And when electrons are lost, they don't just, float away. They are donated to another atom or molecule. So oxidation and reduction reactions are coupled together. So we call those redox reactions. Now, you may have learned some ways to remember this. Does anybody have any tricks to remember which one is oxidation and reduction? Nobody has any tricks? Yeah, that's my favorite one, Christina. So the one that I like is Leo, the lion, goes grr. So Leo is lose electrons oxidation, and grr is gain electrons reduction. And then there's oil rig. Right? The chemists tend to prefer oil rig, but it's not as cute. So oil rig means oxidation is loss, reduction is gain. So you should never forget which one is which. Now, when it comes to biological reactions, oxidation and reduction not only involves electrons, but also hydrogens. So in oxidation, an organic compound loses the electron and a hydrogen. And in reduction, a compound gains an electron and a hydrogen. So for biological reactions in biological systems, hydrogens are also moved along with the electrons. But as we'll see next time, it's all about the electrons. So far, so good? So ultimately, the cell is going to create ATP. So what does the ATP stand for? What's ATP? Yeah, adenosine triphosphate, right? Adenosine triphosphate. And what's ADP? It's not tri, it's di, right? Adenosine diphosphate. So here we have the phosphate groups, and the cell is actually going to remove this terminal phosphate, and that produces the ADP. Now, what kind of bond is it between the phosphates? What do we call that kind of bond? What kind of
Is that a
bond?
valence bond?
Right, a covalent bond, right? And remember, covalent bonds means they're sharing electrons because it's all about the electrons. So when the cell breaks that bond, the electrons can be used, the energy is used. And normally what happens in a cell is the hydrolysis of ATP is a very spontaneous reaction. And so they couple the hydrolysis of ATP with another reaction that's not normally spontaneous. So it's not normally going to happen unless you input a lot of energy. And so the hydrolysis of ATP helps to make that non-spontaneous reaction become spontaneous. And you might recall a delta G gives free energy, and that's what's involved here. We're not going to go over that again. But the whole point is the hydrolysis of ATP is how cells make other reactions occur. So you remove this terminal phosphate, you're left with ADP, which is quickly recycled back into ATP. And the reason for that is the cell uses ATP as an energy source. If the cell had to burn the ADP, it would be left with AMP, and the cell can't do anything with that. So if the cell uses all its ATP and can't make any more, it'll start to burn the ADP, but that's just a fleeting attempt to survive, because if all the ATP is gone, that cell's going to die. So remember, you need ATP to make ATP. Now, we're just going to finish by mentioning the 3 ways that cells make ATP. So the 3 ways that cells phosphorylate to make ATP. And that first way is substrate level phosphorylation. So there's an organic compound that has a phosphate group on it, and that phosphate gets donated to ADP to make ATP. So cells make very little ATP this way. So substrate level phosphorylation can make a little bit of ATP, but that's it. Most cells make most of their energy with the process of electron transport and chemiosmosis, which we are going to review. And so that ATP is made by oxidative phosphorylation. So using an electron transport chain and chemiosmosis results in the formation of ATP through oxidative phosphorylation. And the last way, just to mention it, we are not going to go over photosynthesis in this class. You did learn all about that in 150. There are some microorganisms that are photosynthetic, so they can make ATP using light energy. And that process is called photophosphorylation, right? Phosphorylating using light. So we will mention photosynthesis a few times, but we're not really going to talk about it in any detail. So any questions? This is the easy part, right? The hard stuff comes on Monday. So we are going to stop here for today. We will finish this lecture on Monday. So any questions? So I'm going to shut this down so I can prepare for the lab. So as I mentioned before, lab is short enough today that I will have a lecture that I posted on Monday. I'll actually do it for you today. If you've already listened to the lecture on Monday and you don't want to stay for the lecture again today, that's fine. You don't have to. Okay? So I'm going to shut this down now. I'll see you shortly.